Hello students, welcome to EPG Pathshala. I am Dr. Vishvanath Tiwari from Central University of Rajasthan. Today I am going to talk about a module that is named Enzyme Definition and Characteristic that has been taken from paper Molecular Enzymology and Protein Engineering. In this module, we will discuss about basic of enzymes, its characteristic as well as enzyme specificity. We will also discuss what is the role of coenzyme in the catalysis as well as active site structure. So, in view of all the basic of enzyme, this module is divided into following sections. The first one is what is the definition of enzyme. Then we will discuss what is the characteristic of enzyme, and the characteristic of enzyme will be discussed in the point of enzyme efficiency, enzyme specificity. Within the enzyme specificity, we will discuss what is stereo specificity, what is bond specificity, what is group specificity. We will also discuss what are the other characteristics of the enzyme, and then we will discuss what is holoenzyme, what is coenzyme, what is prosthetic group, and the end we will discuss what is active site and what is the active site characteristics. So, enzyme basically converts substrate into the product, and conversion of substrate into the product is slow. Why it is slow? Because of presence of thermodynamic and kinetic barriers. So, when substrate is converted into the product, there is a existence of a state that is known as transition state. So, you can define transition state as a state which observed during chemical conversion of reactant into the product, or you can say chemical conversion of substrate into the product, which have highest free energy and minimum stability. When a transition state is existing, when substrate is converted into the product, the energy difference between transition state, which is having highest energy, and reactant or substrate is known as activation energy. So, you define activation energy as a energy difference between substrate and transition state. Most of the reaction are slow because the reactant does not have sufficient activation energy which is required to cross the activation energy barrier. So, catalyst as well as enzyme facilitate the conversion of substrate into the product by altering the activation energy, but enzyme is different from catalyst. So, if you compare the enzyme with the catalyst, you can define enzyme are the biocatalyst which have efficiency, specificity and have mode of regulation, but most of the time your catalyst does not have these things. Therefore, enzyme is relatively better than catalyst in view of its efficiency, specificity and have a mode of regulation. As you know that enzyme is involved in the converting reactant into the product via transition state. Therefore, in the graph you can see when a reactant is converted into the product, we are going to have the formation of transition state. Transition state have highest energy as compared to reactant product, and it is going to be most unstable. So, when enzyme is there, that stabilizes the transition state and finally leads to the decrease in the energy of the transition state, which facilitate the conversion of reactant into the product. In absence of enzyme, only few reactant molecule is converted into the product because there are less molecules of the reactant which is having sufficient amount of energy that is available to cross the transition state barrier, and that is known as activation energy. So, in single line you can say enzyme facilitate the rate of reaction by decreasing the activation energy of a reaction. The characteristic of enzyme includes efficiency, specificity and regulation. So, first we will discuss about enzyme efficiency. we have a theory that is known as collision theory. According to the collision theory, the rate of product formation is directly proportional to the effective collision. Effective collision means collision taking place in a proper orientation which leads to the formation of product. But the maximum collision that is taking place in any reacting system in a given time is known as diffusion limit or catalytic perfection. The diffusion limit for most of the reaction will not cross 10 to the power 9 per mole per second. So maximum diffusion, maximum collision that is possible is 10 to the power 9 per second. Most of the uncatalyzed reaction have efficiency in the order of 10 to the power 2 to 10 to the power 3. That means approximately 10 to the power 6 collision are going to be waste. But if an enzyme which is catalytically perfect, then it has efficiency constant very high, or it is close to the 10 to the power 9 or 10 to the power 8, because if the collision efficiency is good, then approximately all the collision that is taking place in a particular condition is effective, and then we have more fast product formation. But in normal circumstances, because of less efficiency of collisions, we have slow reaction, but enzyme facilitate that reaction by providing the environment in such a way that efficiency of the reaction enhances. For example, a catalase enzyme have the efficiency of 3.6 into 10 to the power 7, which is very close to the diffusion limit of any reacting system. Therefore, we can say that enzyme increases efficiency of catalyzed reaction. But from where the efficiency of the catalyzed reaction comes? So enhancement in the efficiency of enzymatic reaction is achieved because of presence of active site in the enzyme. So active site, which is a group-like structure present in the enzyme, creates environment in such a way that most of the collision that is taking place within the active site is going to be effective. And according to the collision theory, it states that rate of reaction is directly proportional to the effective collision. So if active site is providing environment for the catalysis, then efficiency will increase. So the efficiency constant of an enzyme is better than metal catalyzed reaction. Therefore, we can say that enzyme are more efficient than catalyst. Then the second characteristic of enzyme, that is enzyme specificity. When you're talking about enzyme specificity, the specificity means enzyme specific towards its substrate characteristic. For example, the stereo isomer of the substrate, bonds present on the substrate, functional group present on the substrate, or any other property of the substrate. So based on the substrate specificity, we have three types of specificity that is found in the enzymes. The first one is stereo specificity, second is bond specificity, and third one is group specificity. Each of these specificity will be discussed into the detail in this module. First specificity, that is stereo specificity. You know about stereoisomers. So stereoisomers differ from each other in the orientation of groups in three-dimensional space. As you can see here in the structure, we have D glucose and L glucose. D and L, which is written here, is a capital D and capital L. Please don't confuse this capital D and capital L with small d and small l, because small d and small l represents the optical isomerism, where if the light which is fall on the molecules is rotated towards right side, it is dextrorotatory, and if it is rotated towards left side, then it is going to be levorotatory. But here capital D and capital L represents the orientation of group at penultimate carbon. So capital D form of carbohydrates, capital D form of amino acids are those where the functional group which is present at penultimate carbon, if it is present towards right side, then it is capital D form. If it is present towards left side, then it is capital L form. As you can see here in the structure of glucose, the penultimate carbon is the fifth carbon. And in the D glucose, you can see the hydroxyl group is present toward the right side, which shows that it is a capital D form. While in case of L glucose, the functional group is present toward the left side, so it is L form. Similarly, in the case of amino acid, the presence of amino group will decide capital D amino acid and capital L amino acid. But by comparing the structure of capital D glucose and capital L glucose, you can see that they all are identical except the orientation of the group at penultimate carbon. Therefore, if you see that a enzyme which is recognizing D carbohydrate, they are unable to utilize L carbohydrate, which only differ in the orientation of group. Similarly, if an enzyme recognizing L amino acid, they are unable to use D amino acid, which only differ in the orientation of a group. As you can see from the structure of D glucose and L glucose, if you see the structure, then you can say that there is a small rotation of the hydroxyl group is distinguish D form of glucose from L form of glucose. Therefore you can say that enzymes are so much stereospecific, so they can pick a small rotation around a particular carbon. Therefore you can say that enzyme have stereospecificity. If you compare it with the catalyst, catalysts are going to recognize ortho, para and meta form. But when they are producing ortho form, at the same time they are also producing meta form, where the groups are present at different carbons. In an enzyme catalyzed reaction, they are even distinguish the rotation of the bond, while catalysts are unable to produce a specific ortho or meta form. For example, when ortho and meta products are major product for the catalyst, at the same time they are also producing meta as a minor product and vice versa. So stereospecificity will leads to the absolute specificity of the enzyme, and you can define absolute specificity as a property of an enzyme that react specifically with any substrate. Bond specificity. This is the second specificity that is present in the enzyme. When we are talking about enzyme catalysis, enzymes are very specific for its bond on which they are acting. For example, you have heard about peptide bonds. So peptide bonds are formed when two amino acids condense to form dipeptides and tripeptide, tetrapeptide, pentapeptide and polypeptide and so on. So when proteases is going to cleave the protein or enzyme or peptides, they have to cleave the peptide bonds. But when you are using protease, they never cleave any other bond like glycosidic bond, phosphodiester bond or any other bond. Therefore, a particular enzyme is going to have bond specificity that is important for its catalysis. But if you see the chemical nature of the bond that is cleaved by proteases, that is amide bond. So proteases cleaves amide bond. So what happens sometime when we want to design an artificial substrate for a particular enzyme to monitor the catalysis, then we are using the same approach where A particular protease is cleaving a amide bond. So we must have amide bond in the substrate which is cleaved by the protease, but the cleavage product is producing some chromogenic substrates, chromogenic products. Then that chromogenic product is used to monitor the catalysis of that proteases. Similarly, other enzyme, that is glycosidases, which cleaves the glycosidic linkages. Glycosidic linkages are formed when two monosaccharides combine together by removing one water molecule. So if you see the chemical nature of glycosidic linkage, they are ether linkage. So you can say that glycosidic bonds or ether linkage are cleaved by glycosidases. Similarly, if you want to design some chromogenic substrate for it, then you must have a ether linkage in the substrate. So when glycosidase act on that particular artificial substrate, it will remove the chromogenic substrate, and then that produces some chromogenic product which is monitored by different methods like UV visible and other methods. For example, other molecules like lipids which is having ester bonds. So lipases are specific for ester bond. Not only that, the phosphodiester bonds, which is very important, they are cleaved by nucleases, and they are very specific for the phosphodiester bond which is present in the DNA. So if you see that when you are adding protease in a mixture of protein, carbohydrate, lipids and nucleic acid, they will only cleave proteins because they have bond specificity for the protein. If you have glycosidases, they will only cleave glycosidic bond. So if you are adding glycosidases in a mixture of protein, DNA, nucleic acid and lipids, they will only cleave carbohydrates. If you are adding nucleases in a mixture of protein, carbohydrate, lipid and nucleic acid, they will only cleave nucleic acid. So this shows that every enzyme have bond specificity that is important for its cleavage. The third specificity that enzyme have is a group specificity. If you remember any reaction where you remember the structure of substrate, they must have different groups. So enzyme, when they are acting on a particular substrate, they sometimes require a specific group for its catalysis. For example, if you remember enzyme hexokinase, which is or any other kinase, they require hydroxyl group in the substrate. For example, hexokinase require 6' OH group in the glucose. If you remember some protein kinases which phosphorylating the protein, they require hydroxyl group in the protein for its mechanism of action. For example, protein kinases which is serine based, so serine protein kinases, so they will phosphorylate the serine hydroxyl group present in the substrate, that is protein or polypeptide. Threonine based protein kinases will require hydroxyl group of the threonine in the protein. Similarly, some other enzyme require some other groups. So in this way we can say that every enzyme require a specific group for its catalysis. After the discussion of efficiency, specificity, enzyme also have many other characteristics. For example, they undergoes regulation. When you compare an enzyme with a catalyst, enzyme always undergoes regulation. That means if you need a function of enzyme, you can activate it. If you doesn't need, you can silence it. And most of the enzyme we have are proteinous in nature. The important thing why most of the enzyme are proteinous in nature is hidden in the structure of the proteins. So protein can exist in the tertiary folded structure while other molecules cannot. This is the reason why protein is preferred sub molecules which is selected as the enzyme. We also have naturally occurring RNAs as an enzyme that is known as ribozyme. But we have rarely DNA enzymes. The reason is because RNA undergoes folding and can form tertiary structure, while DNA doesn't have folding. It is folded in the form of nucleosome, but DNA itself is not have a folded structure. It is important to know here that when I am saying tertiary structure, folded structure, don't confuse tertiary structure with 3D structure, because everyone have the three-dimensional structure, but I am talking about folded tertiary structure. The other properties of enzyme, they are require a specific pH and temperature for its catalysis, that is known as optimum pH and temperature, and they require a specific substrate with a specific functional group into that substrate for its catalysis, and the affinity between substrate and enzyme is very important, and that is explained by the Km. We also have some inhibitor which can regulate the catalysis. We also have some molecules that is known as allosteric molecules which also regulate the enzyme catalysis. These two inhibitors and allosteric enzyme are discussed in detail in other modules. After discussing the characteristic of enzyme, let's have discuss cofactors and coenzyme. So enzyme itself is known as holoenzyme, which consists of two components. First one is apoenzyme and second is a cofactor. So apoenzyme, as you know, the protein is important component of enzyme, or you can say enzymes are the proteins. So apoenzyme are the peptide part of whole enzyme, and cofactors are non-peptide part of the enzyme. So usually it is a strength of interaction that is responsible for interaction between cofactor and apoenzyme. So based on the strength of interaction between apoenzyme and cofactor, the cofactors are divided into two parts or two types. The first one is prosthetic group and second one is coenzyme. So when you are saying a prosthetic group, then the strength of interaction between cofactor and apoenzyme is strong, while when you are saying coenzyme, then strength of interaction between cofactor as well as apoenzyme is weak. So on the basis of a strength of interaction, it is either cofactor is either prosthetic group if it is strongly associated, or coenzyme if it is weakly associated. So if you see the different cofactor that is available with the whole enzyme, it is either metal, either vitamin, either other biomolecule. So if anything that is strongly associated, then it is prosthetic group, and it is well known that most of the time metal which is present as a cofactor, they are strongly associated with the apoenzyme, that make them as a prosthetic group. So most of the metal found in the whole enzyme, they are behaving like prosthetic group, while organic molecules such as vitamin and any other organic molecule, they are mainly behaving like coenzyme. But please remember one thing, it is not that organic molecule is always behaving like coenzyme. Sometimes organic molecule can also behave like prosthetic group. For example, FAD found in the PDH complex is behaving like prosthetic group. We know that vitamins can act as a coenzyme. So this table shows that how a vitamin is performing a biochemical reaction by acting as a coenzyme. The first vitamin, that is thiamine, that is also known as vitamin B1, its coenzymic form is TPP, that is thiamine pyrophosphate. It is involved in the cleavage and formation of carbon-carbon bond adjacent to the carbonyl carbon. If you remember a reaction where pyruvic acid is converted into acetyl coenzyme and catalyzed by pyruvate dehydrogenase. The first enzyme of that complex is pyruvate carboxylase, which cleaves pyruvic acid and releases CO2. That is not because of carboxylase reaction, but that is because of thymine, which cleaves the bond between carbon-carbon and would remove CO2. The second vitamin is riboflavin, which is also known as vitamin B2. Its coenzymic form is FMN, flavin mononucleotide, and FAD, flavin adenine dinucleotide. They are involved in the oxidation and reduction reaction. Oxidation reduction reaction means transfer of electrons. Oxidation means de-electronation, removal of the electron, and reduction means taking electrons. So, electron or hydride ion. Please don't confuse the transfer of hydride ion with the transfer of proton, because hydride ion representing electron. So, riboflavin catalyzes oxidation and reduction reaction, but they are specific for its substrate, so they require presence of hydrogen or hydride ion in adjacent carbon, then they are going to act. One such example, if you remember, is the succinate of the TCA cycle, which is converted into fumarate by succinate dehydrogenase. Then third vitamin, that is niacin, which is vitamin B3. It is having two coenzymic form. The first one is nicotinamide adenine dinucleotide, NAD, and the second one is nicotinamide adenine dinucleotide phosphate. They are also involved in the oxidation reduction similar to the riboflavin, but they differ in the mechanism because they requires hydride ion to be present on the same carbon. One is directly present at the carbon, other is attached with oxygen or any other strong electronegative atom. So if you remember any enzyme of the TCA cycle which is using NAD, one such example is malate dehydrogenase, which converts malate into oxaloacetate. Then you will recall it that they are going to take hydride ion and creating C double bond O in oxaloacetate. If you compare the structure of malate and oxaloacetate, the only difference is at one position in the malate you have COH, H, and in oxaloacetate you have C double bond O. The next vitamin which is acting as a coenzyme is pantothenic acid, which is also known as coenzyme A, and they are involved in the acyl group transfer, and it is very important when we are talking about fatty acid biosynthesis and its metabolism. So they are going to act as a carrier of fatty acids and acyl group. It is also involved in the transfer of acyl group. That's why it is known as acetyl coenzyme. That is well known molecules. Then fifth one is pyridoxine, which is also known as vitamin 6, and its coenzymic form is known as pyridoxal phosphate. And pyridoxine is a carrier of amino group, and therefore they are involved in the transamination reactions. In the transamination reaction, one substrate having amino group, other substrate have a carboxylic group. So during the transamination reaction, the transfer of amino group and its replacement with the carboxylic group take place. Carbonyl group take place. So one substrate have amino group that is replaced by carbonyl group, and other substrate have amino group that is replaced by carbonyl group, and other substrate having carbonyl group that is replaced by amino group. So basically the transfer of amino to carbonyl and carbonyl to amino is catalyzed by pyridoxine. The next vitamin is biotin, which is known as vitamin B7. Its coenzymic form is biotin carboxyl carrier protein. It is a carrier of carboxylic group or CO2 group. It is very important when you are talking about carbohydrate metabolism. They are involved in carboxylation reaction. One such carboxylation reaction are production of malonyl coenzyme from acetyl coenzyme, and that is catalyzed by acetyl coenzyme carboxylase, which is the first enzyme of fatty acid biosynthesis. The next vitamin is folic acid, and its coenzymic form is known as tetrahydrofolate. Tetrahydrofolate is very important if you remember the nucleotide biosynthesis. They are carrier of one carbon, that is in the form of methyl group, formyl group, or formidino group. So that is important when the synthesis of thymine take place from uracil, which differ only in one methyl group. So the thymidylate synthase enzyme uses this vitamin, this coenzyme, for transferring methyl group. The another vitamin which also acts as a coenzyme is vitamin B12, which involve in the rearrangement reaction of hydrogen atom or alkyl group. And the last and most interesting vitamin is ascorbate, and it is also known as vitamin C, and its function is it is a carrier of hydroxyl group. So if you remember, the stability of the collagen is dependent on hydroxyproline and hydroxylysine. Our body cannot synthesize the proline, which is hydroxylated, or it cannot synthesize hydroxylysine. Therefore, they are synthesized by post-translational modification by hydroxylase enzymes, which uses vitamin C. Therefore, in the absence of vitamin C or deficiency of the vitamin C, the hydroxyproline and hydroxylysine are not synthesized. That makes your collagen weak, and that causes scurvy. Now, we will discuss about active site and its characteristics. So, what is active site? Active site is a group-like region that is present in the enzyme and that is responsible for binding of substrate and any other reactant. Active site consists of two type of residues. One is known as binding residues that are involved in the binding of the substrate to the active site, and the second one is catalytic residues, which are involved in the different type of catalysis that is performed by the active site. There are two models which are given to explain the binding of the substrate to the active site. The first model is known as lock key model, and the second model is known as induced fit model. In lock key model, it is assumed that enzyme is like lock, it has groups, and your substrate is behaving like key. So once a complementary key match with the lock, then reaction will take place. That means once your enzyme which have complementary pocket to the substrate combined with its own substrate, then that substrate is converted into the product. But in the second model, that is known as induced fit model, which is saying that enzyme is not 100% complementary to the substrate, but it is having some complementarity. So once the substrate bind to the enzyme, there are conformational changes take place, and then that substrate is converted into the product. The induced fit model is supported by the results that shows that the transition state have even better interaction than the substrate, and it is only possible when we follow the induced fit model. The binding site residues and catalytic residues that is present in the active site, and they are involved in the catalysis. So the binding site residues form non-covalent interaction such as hydrogen bonding, ionic interaction, van der Waals forces, and hydrophobic interaction with the substrate. And the strength of total interaction between substrate and active site will be decided by these forces. So the binding energy which is produced because of these forces, non-covalent forces, are responsible for selection of a particular substrate over other. Catalytic residues of active site helps in lowering the activation energy of reactant and facilitate the enzymatic reaction. Catalytic residues are lowering the activation energy by following the different mechanism such as acid base mechanism, covalent mechanism, intramolecular mechanism, or any other mechanism, and total attractive forces that is present between enzyme and substrate that are involved in the binding are known as Sarsi effects. So Sarsi effect is like total interaction that is responsible for that binding of substrate with the enzyme. An active site that is consists of binding and catalytic residues remain, regain, restructures once the reaction is over. So that means catalytic and active site residues are important for catalytic function, but active site residues not only playing role in the stabilizing the transition state, but they are also important for the binding of your substrate with the active site. So student, let's summarize what we have studied in this module. So in this module, first we have discussed about definition of enzyme. So according to definition of enzymes, so they are biocatalyst having efficiency, specificity, and have mode of regulation. So then we have discussed what are the characteristic of enzyme as well as what are the characteristic of active site. We have also discussed what is the efficiency of enzyme and how enzyme efficiency is different from catalyst efficiency, and we found that enzyme are far more efficient as compared to the catalyst, and the efficiency in the enzyme comes from specific active site structures. We have also discussed what is a specificity of the reaction. We have discussed how stereospecificity are able to make enzyme to distinguish a particular form of substrate where only one bond rotation are different. If you remember, enzyme are able to distinguish D amino acid from L amino acid. They are able to distinguish D carbohydrate from L carbohydrate. If you remember, then we have discussed what are the bond specificity, how a particular enzyme is going to cleave a particular bond. For example, proteases cleaves peptide bonds. Carbohydrate contain glycosidic linkage which is cleaved by glycosidases. So in this module we have also discussed what is coenzyme, what are prosthetic group, what are holoenzyme. We have also discussed how your coenzymes perform a particular biochemical reaction. At the same time, we have also discussed what is the role of vitamin in the coenzymatic reaction. We have also discussed how a log key model and induced fit model of active site catalysis are playing role in the process of function of enzyme. So in this way we have discussed the basic of enzyme and its different characteristic.